लाइव सर हो गया। हमें मोकले नहीं कहना पूरा कौन होता है।
Jiten, so let me know when I need to share my screen. Okay.
Yeah. Respected Principal Dr. Dhiren Pisa, Professor Dr. Biren Insa, my dear colleagues, and all my dear participants, a very good afternoon to all. I, Jitendra Singh Yadav, on behalf of Vidya Bharati Trust and Sri NARANJIBHAI LALBHAI PATEL College of Pharmacy, welcoming you all in today's webinar. It's a great pleasure for me to introduce our speaker today, who is going to talk to us about quality by design implementation in pharmaceutical industries. He is an accomplished pharmaceutical researcher, over a decade in global pharmaceutical research organizations with a particular interest in new drug product, process development, and commercialization. He has handled new drug product development project for a variety of drug substance from the product design stage through to first time in human studies, clinical supplies, and industrialization. Most of his experience comes from leading scientific frontiers at GSK Research and Development Units based around London, UK. He has been a part of several successful product launches and process validation of the first commercial product on a continuous tableting line. He has also led a product asset swap between Novartis and GSK as a part of business divestment. He also delivered expert talk in many international seminars, conferences, and workshops. Moreover, he is a lifetime member of the Royal Society of Chemists and honorary lecturer in several universities. So, dear participants, let's welcome Mr. Bhupendra Kaushik. So, I request him to take over the session. Bhupendra, sir.
Thank you so much, Jiten, sir. First of all, I would like to thank you to organizers for having me on this session today. I'm going to present on quality by design and how it's actually applied on drug product development. So let me start sharing my screen. Let me know when you see it. Do you see my screen?
Yes, sir. Yeah. It is visible.
Is the PowerPoint presentation visible? Yeah.
Yeah. Okay.
And can you hear me clearly?
Yes, definitely.
Okay. So let's start it here.
Yeah.
Thank you. So today's topic is all about building quality in product, and I will use this term many times, building quality, because that's the intent of today's presentation. The quality by design, the terminology itself tells us a lot about what is actually intended purpose for. So when we actually are developing any product, whether it's in early stage or it's already in industrialization stage or is commercialized, the concept and principle of QbD is equally applied. More emphasis has been put on during the drug development stages because a solid understanding of product and process is must to have before a product can be commercialized. And that is just to avoid future surprises. Okay, so there are going to be some assumptions I'm going to be making today during this presentation. One of them is that most of the viewers today are from the pharmaceutical background. Secondly, I believe that most of us are aware of basic principles of compression and tableting process, because that's what I've taken as an example for. And also an assumption about people who join in today are willing to learn something. And if there are any questions and comments, please do let me know, and I will try to answer them as best of my capability today. So why to listen to me? So a little bit about myself. Actually, I have a bachelor degree in pharmacy from India, Central University, and then a master's in India, and also have done another master's at King's College London. And after obviously my academic career, I started working in pharmaceutical industry. My first job landed in Pune in India as a research associate for injectable product development. And since then, actually, I moved to different companies, starting with QCEPT, where I was doing a lot of preclinical development activities, toxicological studies, testing sort of new products on animal models. And then I moved to MSD, where we did similar things, but this time on humans, first time in humans, preclinical clinical studies. And then I moved to GSK. In GSK, actually, I have done different things, working on inhalation product development, biologics. Last 5 years, I've been involved actively in supporting the product portfolio in HIV and anti-cancer portfolios. And in my current job at Martindale, I'm looking at some of the commercial operation at the moment. So that's a little bit about myself. This is just to tell you that I have actually learned a lot in my 10, 11 years of working career in pharmaceutical industry, done different things. And I'm also always looking to sort of build my career and future by learning new things. So always trying to be in mode of continuous learning. So let's just start with the basics, okay? Um, what we as a pharmaceutical industry and what we as a scientist are facing, okay? The first thing that comes to any scientist's mind when they're actually developing a product is what is the indication this is going to be used for, and if this new medication would be efficacious or not. Because regulators are looking for sort of newer therapies, newer molecules which can work in a novel ways. They just don't want another molecule coming in the market doing similar thing. Okay, so thinking about patients, thinking about the end user, which is somebody which we know, maybe our relative, maybe my own family member. So the first thing that comes to a scientist's mind is this drug going to be safe? So the safety is obviously associated with absorption of the drug. So bioavailability plays a key role. Most of the new chemical compound, most of the new chemical entity, they actually fail at this stage where they don't meet the desired endpoint or don't meet the desired therapeutic effect that we hope that it will achieve. And once we have actually done some sort of preclinical and clinical, let's say phase 1 studies, that is when we actually looking at sort of safety and toxicity profile of the compound. The next thing that becomes quite important is stability of the product. When we actually are manufacturing a drug product for, let's say, phase 1 or preclinical, they are simple solutions or let's say powder filled in the capsules. But as soon as we enter in phase 2, when we actually have to supply to different hospitals, different clinical centers, the volume goes up. And at that stage, pharmaceutical industries are expected to come up with a formulation that is very close to the commercial image of the product itself. Most of the time, it is tablet. And obviously, when we are developing a product is going to come in contact with excipients, it's going to come in contact with container, packaging material, environment, humidity, temperature, and sometimes the active itself can have some impurities in it, which can grow in quantity over a time period. So stability during the shelf life of the product becomes obviously a bigger challenge. Um, there are ways you can actually minimize this sort of impact on product quality, but ultimately, I mean, these are some of the key factors that can affect the success of any product. Okay, and third sort of issue which we facing more mainly in India is cost associated with research and development, because developing a new molecule can easily take up to 10 to 15 years, and that process could be quite lengthy, could be very expensive, because you're going to be conducting a series of development activities, preclinical studies, clinical. So the cost associated is not bearable by all the pharmaceutical companies, considering the size of the companies. So this is just to give you an overview of some of the key challenges we as a scientist are facing in pharmaceutical industry. There are other issues that you may come across during the development of the product, but these are the main ones. And the reason I have put that sort of beginning of my presentation is that is just to give you a flavor of what we're going to handle, how we're going to handle in the topic today. So let's start this journey. For me, when I enter in pharmaceutical manufacturing environment, most of the time I get a call. I get a call from somebody in manufacturing asking me like something has gone wrong in the manufacturing. Can you come down and have a look? It could be sort of simple issue like we have gone sort of or exceeded desired, let's say, sort of range that was mentioned in the batch manufacturing card, or we have gone beyond, let's say, whatever the specification we actually registered the product with, or sometime it could be as simple as that we're seeing some defects during the manufacturing, like capping in tablets, lamination, chipping, things like that. Okay. Um, so what actually happens there, and how do we approach these things? So this is where actually our scientific approach, scientific mindset, a technical, a logical approach kind of comes handy. And this all understanding is simply based on simple science of pharmaceutical technology, which again is dependent on how deeply you understand your product and process. And that is the whole objective of QbD, to develop deeper understanding of your product and process. I'll be repeating this sentence many times during my presentation because what actually we are trying to achieve from quality by design is a consistent product which will be delivering desired therapeutic response every time it's manufactured. Okay, so once a product is commercialized, depending on the volume or depending on the commercial demand, you may be manufacturing more than 100 or 200 batches every month, maybe every year. And how do you ensure that every single batch you manufactured achieves the primary objective of this product, which is obviously treating the patient, or also, let's say, meeting the desired shelf life of the product, meeting the release specification, obviously during the product testing, and also the shelf life. So thinking about patient safety, we are more concerned about patient safety. We are concerned about product quality, efficacy, and safety. And by focusing on building the quality right during the development, through the industrialization and commercialization, we make sure that every single product, every single batch we manufacture meets the desired specification. Okay. And that is the ultimate objective of this quality by design principle. And how do we achieve it is something we're going to discuss later in the presentation. So there will be two things which we'll be talking a lot about in this particular topic is finding your design space, or let's say designing your design space, and also output of that design space is actually summarized in product control strategy. Okay, so we're going to go into that. Let's start with the design space. Okay, in my slide here, what we have is a simple picture of let's say this control space, which is this small orangey area, followed by this yellow area, which is our design space, and then the greenery space is your knowledge space. Okay, so let me tell you more about these three spaces here. When we are studying pharmacy, we are sort of trying to get understanding of basic principle of pharmaceutical technology, science, different topics, analytical chemistry, medicinal chemistry. It just gives us theoretical understanding. Okay, but that knowledge is something that you can use in your knowledge space, because as a scientist you've been given this education and that is what you've been trained for. Okay, when it comes to design space, this is where you start asking question to yourself, like, do you understand your process? Do you understand your product? And when you answering these questions, you start going into detail. Okay, what if things go wrong? What are the things that can go wrong? And if they go wrong, how does that impact the product quality, efficacy, or safety? How likely is that to happen? And if that happens, what are the key risks and how we can minimize these risks? And that is the basic concept of quality risk management. Okay, but this design space here is purely to tell you that in order to establish a deeper understanding of your product, you're going to have to carry out a series of experiments. you're going to have to design certain experiments that would allow you to answer some questions which actually are already sort of output of a scientific thinking. Okay, so we're going to come to the design of experiments and how we actually sort of interpret those results later during the presentation. Our focus here is finding the control space. What we're saying here in the control space is that within that small boundary, your product is quite stable. Within the design space, you understand that there may be certain variation during the manufacturing of the product, but you understand its impact. And in the knowledge space, what you're saying is that based on your knowledge and previous historical, obviously, experiences, you may be able to tell that to how this process can affect or process variation can affect product quality. Okay, there is very subtle difference, but knowledge space is purely theoretical. Design space is based on your experimental studies with the purpose of finding the questions you actually want to answer, and control space is what you actually register for product launch. Okay, so when you are registering your product for launch, you are expected to provide certain limits or ranges for critical quality attributes of tablets. Okay, so critical quality attributes, obviously for a tablet product, let's start with an example. Okay, for most of the tablet, there are mainly 6 critical quality attributes. They start with description, identification, assay, which is simply the drug content, uniformity of drug content, which is the distribution of drug content, which could be plus minus 5 or 10% depending on the tablet size. And then actually you have dissolution. Dissolution is simply mimicking the response of how once the drug is actually inside your body, it releases the drug. Okay, the rate and extent of it in a human body basically sort of gives us the bioperformance, which is linked to the bioavailability. But every time we manufacture a batch, we cannot dose the product to humans because it's just too expensive. So what we do actually, we come up with an in vitro model. In vitro model is a dissolution basket where you actually dropping your tablet from a batch or batch you manufactured to compare how that profile of drug release matches with bioperformance which you carried out during your phase 1 or phase 2 study. And that is how you try to do the in vitro in vivo correlation, and that falls into the category of critical quality attributes. Last but equally important is impurity profiling. As I said, during the development of product, when you are manufacturing your active in a smaller batch, you may see sort of different impurities which may come from the synthesis process itself. But during the scale-up process, your impurities may also come from the product contact parts. Okay, like if you're using silicon or stainless steel surface, your active may interact with it. Okay, there's possibility there. And this all actually awareness helps us to actually understand our product. And as I said at the beginning of this presentation, the whole concept is all about getting a deeper understanding of our product and process. So that is just to give you sort of flavor of what we're going to be targeting today and how we're going to target it. So I'm not going to go into definition of CQAs, control strategies, and designing space. I mean, it's already out there, but we're going to look at some examples today, which will then give you a deeper understanding of why we're studying certain things and how we can study those things. Okay, so let's start. When we actually have a new product, okay, and when we are asked to understand it, okay, there will be places where actually you can say like, I have studied it, or we have manufactured this product or similar product on this site, and we actually understand it. Okay, so that is based on your sort of historical understanding, which is okay, which is acceptable. But there will be areas where you will be asked questions. Okay, what actually may happen if you change, let's say during the granulation process, if you added too much water. Okay, then you may say like, okay, I can kind of tell that this may affect the drug dissolution, but I don't know to what extent. Okay, then you may be asked to perform an experiment and demonstrate this practically of the extent of impact on the product quality. And this is where designing the experiments come in place. The design of experiment is literally looking at one or more variables and impact of those variables on one of the CQAs. okay, critical quality attribute. Most of the time, the CQAs we are targeting is dissolution because that is very closely how we actually see the performance of product in a human body, okay? And what we have to then actually do is evaluate our experimental data, study it, interpret it, and make some conclusive sort of report from it. And that is also obviously responsibility of a scientist. So there is actually quite a huge sort of, let's say, responsibility on a scientist's shoulder to be able to sort of do these experiments, do this gap analysis, do this risk assessment, and then actually design your study and come up with some sort of data which you can explain to regulators that whatever we have done here gives us more confidence on our product and processes. That is the ultimate objective here. So the different stages here are actually designed in a very scientific and a particular way to give you the answer you're seeking. Okay, so let's just start with the understanding our active itself. A lot of the time, which we sort of understand that our active play a crucial role in how our product is going to be performing in the body, which is simply means is by performance. But there are other things which we don't normally pay attention, could be equally important, is physical chemical nature or physical chemical characterization of the active itself. Okay, so when you look at this table here on the left, what I have here is let's say simple things like how much water is in your active, how much solvent is in your active. The solvent could be methanol, acetonitrile, which is used during the processing. And what will happen if we change the shape and particle size of the active, okay, which is looking at the X10, X50. This is data from particle sizing using the laser diffraction technique, and also the impact of change in tablets, well, sorry, actives bulk and tap density. There are different variables, right? That plot kind of tells us that there are lots of variables in active itself, okay? In order to establish impact of each of those variables in your product, okay, when you use this active in your tablet, then it becomes a product, then how that characteristic, how that attribute of active itself can affect the CQA, or let's say the performance of your tablet. Yeah. So on the right side, what we have here is listed CQAs on the top here, and on the left side here on the y-axis, let's say on the first column, we have some drug substance attributes. So on the first row, what we see in here is the solubility of the active itself. Okay, if the solubility of the active is poor, it is very obvious that it's going to affect the dissolution. Okay, if you have higher, let's say, water content in your active, okay, and if that water doesn't get removed completely, then actually that may affect it, but likelihood of that happening to different CQAs, I would say is low. um, there are other things which is around nature of the active itself. They could exist in different crystalline form, polymorphic form, and different polymorphic form or amorphous form of the active can have different solubility profile, and that is how they can also affect the dissolution of the compound of the product here. Okay, so these two sort of data set here is telling us that the amount of variability that can come from active itself is huge. Okay, and if we don't study them well, we basically are not preparing ourselves for future, because during the life cycle of a product, you may get good quality active coming from, let's say, smaller scale manufacturing during the development, but when you go for scaling up, when you go for manufacturing your active from, let's say, UK, US to China, Brazil, India, places like that, you may see sort of different profile, just because the product now is manufactured on a different scale, different obviously manufacturing equipments, manufactured by different processors, different people. So certain attributes of active may change. And as a scientist, again, it's your responsibility to be able to look at this data or look at this theoretical risk assessment and then come up with some sort of plan to say, okay, we understand this risk, but what we can do to minimize this risk, to mitigate this risk, or how we can actually avoid it. Okay, so there are things that you are expected to do, but this information here is only to tell you that there are variables that would come from your active. Let's go for variables that comes from the process as well. So in characterization of an active here, we use a lot of analytical tools. Okay, we use a range of techniques. So this is just for a smaller molecule, and this is just to tell you the type of analytical expertise you expected to understand and interpret. I mean, the first one is simply looking at the PXRD, which is X-ray diffraction, and looking at the solid state properties of the active. The second plot is looking at DVS, just the moisture absorption pattern of the active. Third one is just looking at the shape and size of the active, and different techniques they get used to characterize this active. So again, whatever you're studying today in analytical sciences will be actually applied to this sort of challenges. So whatever you're studying today, please pay attention to it, because in future as a scientist you expected to run these machines, interpret the data, and make scientific conclusion out of it. The complexity here is relatively simple when you compare this against biologics, because in order to characterize a large molecule or protein peptides or molecular antibodies, you expected to run at least 20 different analytical techniques. And that is just to study the activity and stability of this compound. Okay, the idea here is to tell you that you will be expected to know analytical techniques, and depending on the molecules or the field you're working, you may have to adopt your technique. Okay, but these sort of techniques again are used just to study your active molecule. Yeah, just keep in mind. So the complexity here obviously is different in different functions, different organizations, and depending on the availability of the equipment. But there is expectation there. When we go into the process part of it, this is just an IPO diagram. I call it IPO because what we have here is input on the left here, and then in the middle you have your processing, and then on the far right here you have your output. So this diagram is simply telling us what are the inputs that's coming to this process. And on this occasion, I'm using granulation process as an example. And then what are the outputs that's expected of this process. Okay, so what we have here is a product which is using microcrystalline cellulose, sodium starch glycolate, povidone, water, and the granulation process here. Okay, your variability can also come from your excipients. For an example, the particle size distribution of povidone can affect obviously the preparation, the waiting time of the granulating media itself. Okay, in the middle you have your controllable parameters, which are the process parameters that you may have opportunity to change. And also during the design of experiments, you may be stretching them from one end to another end to generate more data. For an example, simple things like order of addition of your active in the granulation process can also affect product quality, as simple as that. The amount of obviously the water you added, the positioning of the spray nozzle, the length of the spray nozzle, the impeller speed, the chopper speed, and the type of recipe you're using to granulate the product. These are variables that are coming from your process. Okay, in the first few slides we saw the variables coming from the active. What we look in here is the variable that is coming from your process. Okay, granulation process is simple. In a way, you have your active with excipients mixed, added some water or granulating media, and then you actually have created some sort of weight mass, and that weight mass get transferred to a dryer. Okay, but look at the number of sort of variables here, the possibilities of things that can go wrong during the process. Okay, and that is the whole intent here. And depending on how you have actually controlled your process, your ultimate output can be impacted. And what is the output of a granulation process? It is simply a granule which you want to keep consistent. So in order to measure it, that it is consistent, you can look at certain attributes like density, its shape, particle size distribution, porosity, water content, things like that. Okay. And why do we measure these things? Because you want to ensure that every batch you're manufacturing is consistent, is going to be manufactured according to the process we registered, is going to be manufactured in accordance to the batch manufacturing record you given to an operator. And also, these are the things that affects or can affect the product quality. So obviously you want to have certain degree of control. So the control you hear or studied, developed, or sort of let's say recorded is actually summarized in product control strategy. So we're going to come to that product control strategy in a minute. But idea here is just to give you sort of level of complexity which you may come across when you're looking into sort of designing your process as well. Okay. So first part was to understand how the variability can come from your active, a little bit about how the variability come from your excipient, and how the variability come from your process. This sort of study can be done using different tools. I have used here IPO, which is input, processing, and then output. But equally, you can use Fishbone, looking at 5 amps, and 5 amps are simply looking at, your reliability may come from your manufacturing operator. Maybe the operator is not trained enough. Maybe the operator is trained, but that day he's distracted. Okay, human factors. And maybe your procedure is not clear. Maybe the material you used during the processing of a particular batch was not of good quality. Okay, your measurement technique, when the machine gets older, the performance of the machine also changes. When you're changing the machine from one unit operation to another unit operation, or one area to another area, the performance can change. Okay, the type of control that you need is just, there are lots of things that can go wrong. And in order to brainstorm, in order to visualize, in order to understand or record the number of variables, you can use these sort of tools. Okay, and once you have identified your process variables, you can start generating this type of transformation plots. And transformation plots are simply are pictorial presentation of what happens when you are overweighting your granule, let's say. What if you are underweighting it? So these sort of tools are actually just to give you more mechanistical understanding of things that can affect your product's quality, efficacy, or safety. And there are like more than 15 different tools mentioned in ICH Q8. If you're willing to have a look at them, please go ahead. Um, what we have actually here, sorry, let me just go back. What happens ultimately once we have done this all studies, your data can be presented in several ways. One of the way here is a surface plot. Okay, our brain can't sometime understand three-dimensional pictures. Okay, but just to keep the thing simple, what we have here in the first graph here is looking at impact of change in concentration of excipients on rate of dissolution of the product. Okay, so A is your microcrystalline cellulose, B is obviously your PVP. So what is telling us that, looking at the direction of the plots, the small changes in the quantity or quality of your excipient can also affect the rate of dissolution. What I have here is multiple variables on the right side, where we're looking at the thickness of the tablet, the amount of work done on the product during the granulation process can also ultimately affect the dissolution process. So what these two plots are telling us that your variability obviously is coming from your excipients and is also coming from your process. Okay, so the granulation process, thickness, and ultimately is affecting the CQA, which is dissolution of the product. Okay, as I said during the presentation, at the beginning of the presentation, that we focusing on dissolution here because that is one of the attribute very close to how this drug is going to perform inside a human body. Okay, and that is the whole idea of in vitro in vivo correlation. I'm repeating myself, but this is very important to know, because in order to generate a fruitful data, in order to generate a useful data, it's important that you pick the right variables and you pick the right measuring response. Okay, I'm going to go to another slide here which will give you more idea about it. Just a second actually. So this is kind of linked to that process where what we have on this slide here is we actually are looking at this response dissolution, as I mentioned. What we actually monitoring, measuring is attributes, which is granule density, size, as I mentioned in the IPO diagram. And what we actually playing with or sort of stretching is the water amount and weight massing time. Okay. And this sort of designing of experimental data is called attribute based design space. Okay. Because you are monitoring your attribute at every stage. When you are going from a smaller scale to larger scale, when you're going from one unit operation to another unit operation, when you're manufacturing your product in one site to another site. And by doing this sort of studies, you can develop mechanical understanding of how your product is going to perform when you change certain process parameters. Okay, so what we have seen here so far actually is literally different tools we can use to understand the process variables, how this data is plotted, and how actually we can use attribute based design space to generate the fruitful data. The summary of this all data actually is actually summarized, let's say, in the product control strategy. But before we discuss that, I would like you to start thinking about how in a simple process, which is blending process here, you will be asked to find, it may happen in your interview as well, you may be asked to find what are the process variables that can go wrong. So this is just a question just to get you thinking that a simple process like this, where you have your granule with some extra granular adds and some lubricants, mixing obviously for a specific time, and also mixing with a specific RPM. So your mixing time and mixing speed. What also is important that the angle of this mixing process, because the way your drum is angled, obviously parallel to the machine, or let's say parallel to the floor, can also affect the performance of the mixing process, because ultimately you want to achieve a uniform product. And in order to obviously achieve that uniformity, you need to mix it for sufficient time and for, let's say, sufficient RPM. So small things like that, small things of processing can affect ultimate quality of the product. And this is just to get you thinking, okay? What we actually then do is, once we have obviously identify our critical process parameters, we start looking at how those process parameters or process variables can affect the critical quality attribute. So on the right side, what I have here is looking at the listed CQAs, which I mentioned earlier, obviously identification, description, impurities, dissolution, content, uniformity, and content. Okay. What I also have here is input variables in the first column, and then moving from your input to granulation process, drying process, milling process, blending, ultimately compression and coating. Okay. There are some areas which are obviously color red. There are others which are colored green, and there are others which are colored yellow. And they mean different things, okay? So for an example here, as I said during the presentation, that your particle shape and size, okay, can affect solubility of the product. And that can also affect the product density or granule density. And that can ultimately affect the tablet dissolution. Okay, let's take another example. During the compression of the product, the amount of pressure applied, okay, can affect obviously the rate of dissolution. But also during the compression process, you need sufficient mass inside the dies to build the or make the tablet, and that actually controls the total amount of the drug inside the tablet, and can also affect the uniformity of the product during that processing. Okay, so during the compression process, this particular step can affect CQAs here. And another example is literally looking at drug substance identity. I mean, if you don't do your identity test, you may be using a substandard active, or maybe using a completely different active, which probably you're not registered with, okay? So what we have demonstrated here in this control strategy, or the picture here, is actually how certain material attributes and process parameters can affect your CQAs. And this is basically summary, and this is how we actually record our product control strategies. And each of those unit operations or each of those processing parameters can have ranges, because within a tight range, within a particular range, or within that particular distribution, your product is in control. But once you've gone outside that range, the performance of your product may be affected positively or negatively. And that comes from experimental data. And that is where your design space plays a crucial role. Because as I said, you have your area, small area, orange area, if you remember, that is where your product is registered, and that is what these all ranges are all about. But there's a design space where you actually can generate the data, not necessarily register it, but at least you understand your product performance. Okay, so this is our product control strategy, and the ultimate objective is here to give you control in your manufacturing, that control you apply in every single batch, every time, so that you meet the desired therapeutic response, safety, efficacy, and quality of the product. Consistency is the word. The ultimate objective of QbD is to deliver a quality product every time you manufacture it. Okay, that is what we're trying to achieve here. So what we done so far here, we looked at CQAs, CPPs, manufacturing variables. We done some physical chemical characterization. We looked at the formulation. We looked at the analytical variabilities. What we also have is looking at other products on the side, looking at other processes on the side, looking at cleaning strategies, regulatory strategies, also trying to assess the risk coming from other areas. You may be manufacturing, obviously, more than one product on the same site. There may be a risk of cross-contamination between the product if you're using the same equipments. And also there's a certain degree of risk sort of management involved, because you cannot study everything. Let's just be honest, okay? You cannot study every possible thing, or you cannot predict every possible avenues. So what you do here is a theoretical assessment, again, using the risk management process to say, what is that risk? Do we understand it? If we don't understand it, then do we need to generate data? And if we cannot generate the data, can we use scientific understanding to justify why the risk is low? Why do we think the chances of this getting detected is okay? Detectability of it, obviously thinking about the ultimate impact to the product quality, and then how likely is that to happen? Okay, so you can use different approaches again to get that sort of recorded theoretically, or ultimately you may be asked to generate more data. All right, so we already have talked about this slide. I'm going to give you some example here of process optimization, and this is just to get you thinking again that the process variability doesn't stop. Okay, the amount of variability that may come is just, it just, there are things that certainly you would only see when you actually experience it. And what I have here is a PMA 25, which is a smaller scale granulator, moving to a medium sized granulator, PMA 65, and then moving to a PMA 150, which is a larger granulator. So when I say larger and smaller, which simply means a smaller batch here, a medium sized batch here, and a larger batch here. Okay. What I was seeing in this particular example is when we were going from smaller scale to larger scale, the performance of the product was changing. So maybe for this scale, this amount of water and this amount of wet masking worked. Okay. But when we were moving to larger scale, there was some impact on the rate of the dissolution of the product. Okay. And then when we actually went to sort of bigger batch, then again, there was impact on product performance here. And the complexity here is that this information could be relevant and could still be useful for this size batch, but may not be so relevant for this size batch. So your variability can also come from equipment, the size you're manufacturing, and where this, obviously the age of the equipment itself, because same equipment was performing slightly different 10 years ago when it was new. Okay, after 10 years, performance may change. So you may have to adopt your strategy. You may have to generate more data. You have to do more experiments to come up with a sweet spot, or let's say an area, or where you have more confidence that every time we manufacture within that space, whatever we will produce will have the desired effect. Okay, and this is what we were seeing, that when we were actually manufacturing the batches at different scales, at different, let's say, processing parameter of water, weight massing time, the rate of water addition during the granulation. Our bioperformance, again, this is taken from dissolution study. What we were seeing is there wasn't much of a difference in terms of dissolution rate, but there was subtle difference, okay? So the difference could be significant, but not necessarily can affect the bioperformance. And this is where, as a scientist, you got to be thinking that, okay, so I have some data here, and I have demonstrated here that, okay, there may be some variability, but that may not ultimately affect the overall performance of the drug, okay? This is what we want to achieve, a robust product where smaller changes to processing parameters does not affect the overall performance of the product. So this product is robust. Small changes doesn't affect the product quality, efficacy, and safety. And that is what this quality by designing is trying to achieve, building the quality at every stage of manufacturing. We have studied every single process parameter. We have thought of every possible avenues. We have also thought about the things that can go wrong from input, from processing, and at different stages. And the way we're studying, we applying a scientific, rational, a scientific approach to it by using design of experiment, different tools, and then generating data, which will be giving us some sort of confidence. So ultimately, what we're doing here is moving from traditional approach to a futuristic approach. And it's all about going scientific. It's all about understanding the variability. And whatever we're doing is all knowledge and data driven, instead of saying, oh, well, actually, this will not affect it. But no, let's do some work, and let's look at the data and then make a decision. Okay. And that is what ultimately QbD is asking for. Like let's think about everything that could possibly go wrong. Okay. And then when you have generated this data, that gives more confidence to the regulators that you actually have demonstrated by data that you understand your product, you understand your input, you understand your processing variables, you understand your product, right? So ultimately, you have sort of proven that you if there are things, if there will be things that may go wrong, it may go wrong, but you will still be in control because you will be able to tell whether it's going to have positive or negative impact in the overall quality of the product. Okay, and that is what we're trying to achieve. So that brings to my last slide for today's presentation. Ultimately, what we're trying to do here is obviously looking at product quality attributes, trying to sort of come up with some sort of specification, come up with process parameters which are critical for the product, and trying to define the process controls that we could put in place. So at every stage of manufacturing, if you are within that range distribution, then everything will be okay. And if you go beyond that, then we can use scientific approach or risk assessment approach to say why we should still be okay. And that helps throughout the journey of the product, right? Because the issues doesn't stop when you are manufacturing the product at a smaller scale. Maybe a lot of impurities are coming from your active, but when you go for a larger scale, maybe the impurities are coming from your processing equipment. And when you are actually are manufacturing your commercial batches, batch size can affect the overall quality. It also dependent on, let's say during the product launch, you manufactured this product in UK, but during the life cycle of this product, you moved it to Brazil because it was cheaper to manufacture there. Okay, but they were using different equipment, so their process control may be different, right? And their performance may be slightly different. But if you have a solid understanding of product, then you can suggest, okay, actually we are seeing these changes here in the product performance. Can we tune this parameter a little bit here and there? Let's change the hardness. Let's change the friability. Let's just change the granulation parameter here. And by doing that, you're actually trying to tune your product to the required quality specification. And more the data you have, more the data driven you are, and ultimately you demonstrating a robust understanding and giving more confidence to the regulator that you are in control of your manufacturing and you understand your product. That is all about my presentation today. Hopefully, I have given you sort of flavor of things that can go wrong, and also how do we actually identify them, I've given you some tools. I've also given some example of how you can design your experiments. And what I've also tried to do is actually give you some sort of idea of how you can use your scientific approach, which can then be data driven, and also give confidence to the regulator that how you use this data to sort of tell them and demonstrate them that you are in charge of product, you are in control of the product, and whatever you're producing here is of right quality, efficacy, and safety. That is the ultimate aim of the QBD. And I hope you enjoy this presentation. I'll be more than happy to take any questions if there are any.
Thank you, sir. Thank you, Upen, sir.
Thank you, Jiten, sir. This is a great platform, actually. Thank you.
Very nicely explained, sir, the process parameters. And I have few questions for you. Actually, that is only the point of view regarding the M-Form students and all.
Mhm,
That if suppose we have think to apply a design,
mhm.
and in the starting, very basic, that
Yeah,
if we have to select a parameter, mainly it is important that we have to go for the some pre-formulation study for that, or on a trial error basis we can put the limits of upper side or lower side like that.
you can actually, because what we have said here is that maybe on the same manufacturing side, you may have more than one product, okay, and you can use a rationale to say that we have similar products on site. We have actually sort of similar processes on site, which we have used, because in a tablet formulation, for an example, most of the process parameters are nearly the same, okay? Most of the formulations are the same. So the type of impact you may see because of change of your active is kind of predictable most of the time. But again, depending on what you're looking for and what is the risk, you may adopt your approach. So if you are concerned about an impurity that is generating during the processing of the product, you may have to apply slightly different approach than knowing that there's already an impurity in your active itself, and that active is that impurity is stable. But if your impurity profile start changing during the manufacturing, or let's say the physical chemical nature of the active starts changing during the granulation process, you may have to adopt your strategy to it.
Yeah. Okay. Okay. And again, one point you have specified during the lecture, the addition of water time during
Hmm.
granulation will affect the product.
Yeah,
So whether the pattern by which we can add water, that also maybe affect the product. Suppose we have
exactly. So
got a time that in 3 minutes we have to add 1 liter of 1 liter of the granulating fluid. So
mhm.
sometime if at once we can put half liter and then the remaining time we will put remaining quantity, it will affect the product,
It will,
the pattern
because
by which we can adding the granulating fluid.
it will, because what happens in a granulation process is basically different excipients coming in contact with water, okay, and the rate they come in contact with water is important, because some of the excipients could be water loving, okay, they would probably get absorbed quickly, and others they may take time. But if you have added too much water too quickly, then maybe the distribution of water may not be uniform, and that is where the rate of addition of water plays a crucial role, and ultimately can impact the ultimate quality of the granule.
Yeah. Yeah, Bhupen sir, actually very nice session. Okay, and one point, angle of mixing equipment from the surface, that actually that will also affect means that how we can fix the equipment and the angle. It will affect
Exactly.
drastically or it will affect in a lesser minor.
So during the validation of an equipment, is actually asked to fix the angle of rotation. It's usually 30 to 45 degree during the mixing process. So most of the equipment, they have fixed angle. But when you actually are moving your product from one equipment to another equipment, that angle may be slightly different. and depending on the volume of your product inside the blender,
Yes, yes.
it can affect the tumbling process inside the blender itself, the drum. And if the volume is not changed
Oh,
dramatically, and also depending on the dose of your product, if it's a low dose product, they're at high risk compared to the large dose product. So it depends on a number of things. Obviously, as I said, it depends on the dose. It also depends on if you using the same equipment and the overall fill volume of the drum as well.
okay. Okay. Thank you, Bhupen. It's a very informative session. And for the M.Pharm student who are doing the projects and those who are applying the QBD in their formulation development, definitely they will help with this particular webinars. There is one small announcement for the participants that don't worry regarding the certificates, and after the webinar you will get a link for the feedback form. You just fill the feedback form, and as soon as we receive the feedback form, we will release the certificate of you. And now I hand over the session to Dr. Dikshit Modi sir for the vote of
Thank you, Jitin
thanks.
sir.
Thank you, sir, for your sharing a wonderful session. Good afternoon to all. Myself, Dr. Dikshit Modi, Associate Professor, Sri Naranjibhai LALBHAI Patel College of Pharmacy. Today, it's my privilege to share a vote of thanks to those who have directly and indirectly contributed to this webinar on quality by design, implementation, and industry perspectives. First of all, I would like to thank to our today's distinguished speaker, Mr. Bhupendra Kaushik, for giving such an inspirational, wonderful, and knowledgeable presentation on quality by design and make really this webinar very interesting and knowledgeable. I would like to extend my thank to our management of Vidyabharati Trust, all the trustees for continuous encouragement to arrange such type of webinar in pandemic situation. I extend my deep gratitude to our principal, Dr. Dhiren Shah, for his continuous support and presence throughout this webinar. I also extend my special thanks to today's webinar coordinator, Dr. Jitendra Singh Yadav, for arranging this webinar. We are highly grateful to Mr. Bhavesh Patel for providing this IT platform for smooth conduction of this webinar. Also, thanks to all staff members for their support and making this event fruitful. Without participants, any event is not possible. So finally, I am highly thankful to all participants for their active participation to make this webinar successful. Once again, thank you all for your support and presence. Thank you also to Mr. Bhupendra Kaushik for wonderful session. Thank you, sir.
Thank you so much. Pleasure is
Thank you very
mine.
much,
Thank you.
sir. Thank you very much, sir. Thank you, Bhupendra, sir. Yes.
Thank you,
Bhavesh, sir,
sir.
please.
Actually, thank you so much to everybody who.
Hello, Bhavesh, sir.
हाँ सर हेलो
Bhavesh, sir. Bhavesh, sir. Yes, yes, yes.
हेलो स्टॉप
Jiten sir, sorry, actually I had some issues with my connectivity.
Hello, Bipin sir.
Yeah, I can hear you, sir.
You are online. Actually, just the host have given the screen full screen for that
Sure.
particular video, and just after that we just
That's okay.
yeah, just few minutes. Yes.